Hi class, Dr. Jim here. Chapter 14. We are now getting into immunology. Now, I could spend days and days and days talking about immunology, but we're only going to cover it in the next two chapters. Really, the next three chapters. We're going to look at the innate immune system in this chapter, chapter 14, which is really what we consider non-specific immunity. And so we'll talk about why that is non-specific. Then in chapter 15, we're going to talk about the adapted or sometimes the acquired immunity, which you develop over your lifetime. This is going to be specific. And so this one targets very specific things. So you need specificity, you make memory, and you do all these different things in the acquired immunity. And so we'll talk about that in chapter 15. Then chapter 16 is going to be looking at what happens when the immune system goes wrong. And so we'll look at some of those different things where the immune system goes wrong, where you have allergies, sometimes you get tissue rejection, sometimes you have antibody antigen interactions that are not good with the blood and other things like that. And so we're going to look at all these different things in chapter 16. So today what we're going to be focusing in on is chapter 14, which is the innate immunity. And innate immunity, like I said before, is non-specific. That means it doesn't matter if it's a bacteria, a virus, or a fungus, it's going to attack it the same way every time. Okay. And what we really concern ourselves with are really the first two lines of defense with the innate immunity. We look at mechanical barriers, which is the first line, and then the second line is your white blood cells. Now, it's not all the white blood cells, but really the ones that are responsible for phagocytosis, inflammation, and then also activating complement. So we'll take a look at all these different things today that are associated with the innate immunity. Okay, so let's get started with this process and look at chapter 14. So the first thing we're going to look at is what is immunology. And like I said, I could spend days and days and days talking very specifically about immunology and all the different parts. People do specific research looking at immunology and spending a lot of time doing the research on that. And so we'll talk briefly about what is immunology and that. We'll also look at the immune system. Again, we'll talk briefly about what the immune system is. And again, it's your defense system against the foreign invaders. Okay. We'll talk about the three lines of defense. Again, the three lines of defense in this case are going to be the first line, which is your mechanical barriers, your second line, which is your white blood cells that kind of sit below the surface, and then finally it's going to be your third line, which is your acquired, your B and T cells. So, and that will be chapter 15. And again, how does each one act? We'll look at what are the mechanical barriers. That's the first level. And then the second level is looking at phagocytosis and inflammation. So what do those white blood cells do in that second line of defense that really add to your innate immunity? Okay, so that's what we're looking at today. All right, so let's go on. So what is the structure and function of the organs of defense and immunity? So anytime we're talking about the defense properties of the human, we're looking at immunology. Okay, and so that is the study of immunology, looking at the second and third lines of defense specifically. And again, what does the immune system do? The first thing that it does is it keeps a constant surveillance. So think about this. You have these cells going around your system that act like security guards, and they're walking around kind of like a security guard would, looking to check to make sure that everything is normal. If things aren't right, they respond and then they react. And so that's what your immune system does. Once it recognizes foreign material, it goes in and then it destroys. It goes in and whammo, it takes out the foreign material. So we'll talk about what is foreign versus self and all these other things. How does it recognize these things? And so this is all a part of immunology. So one is surveillance, two is identifying, and three is getting rid of. Okay, and so that's what we're going to be looking at today. And again, all these different systems interact with one another, and we'll talk about these things as we go along throughout this chapter. Okay, I wanted to start off with a few immune cell definitions, and we're going to be talking about these things over and over again. That's why I bring them up here. One of the big things is the white blood cells. The white blood cells are the WBCs, and again, these are the second types of cells that we have in our blood, and so they're outnumbered by far. So most of your cells in your blood are your red blood cells, but the white blood cells make up about 1 to 2% of your blood, and these are responsible for fighting infection. And so these guys are there for surveillance, destroying, and acting upon foreign invaders. So we'll look at these things. And the other name for them are called leukocytes. So we have the red blood cells, which a lot of times are referred to as erythrocytes. The white blood cells are referred to as the leukocytes. Okay. The lymph is the fluid that leaks out of our vessels. So you might think that our system is a closed system, and we talk about this in A&P a lot, that our blood system is a closed system, that you have arteries leaving the heart with the blood. It takes it back, it takes it in the capillaries. The capillaries then go lead to the veins, and then the veins lead back to the heart. And we call that a closed system. However, our vessels are very leaky, and what that means is that fluid leaks out of our vessels all the time. And there's a reason for this. It's not that we're just made poorly, it's because we want to make sure that fluid, nutrients, and cells get to those areas that aren't touched by the blood vessels themselves. And so these are important. So it is okay to have leaky vessels, and we'll talk about that. When the fluid does leave the vessel, we call this lymph. And lymph is the extracellular fluid that eventually returns into circulation through the series of vessels called lymphatic vessels or lymphatic channels. And we'll talk about that as well throughout this chapter. Another thing that you're really going to hear, probably not so much in this lecture, but in the next lecture, is the difference between non-self and self. Non-self is anything that's foreign, that's not supposed to be there. Self is anything that's supposed to be there. Now, we have markers on all our cells except our red blood cells that tell us that this is us, don't attack. And so we'll look at that in chapter 15 and then again in chapter 16 when this doesn't work. And so that's the difference between non-self and self. So we're looking at all these different things today in that and kind of giving you an idea of what these things are. All right, so we're going to look at the host defenses. And again, we can break it down into two main systems. The innate system, which is your natural defenses. These are what you're born with, okay? As soon as you come out of the womb of your mother, you basically now have these innate defenses that can attack anything that comes into your body. Now, these aren't the best defenses in the world, but they do keep you pretty healthy throughout your life, okay? And so these things are non-specific, and they'll attack viruses, bacteria, fungi, parasites the same way that they would do any other organism that they find foreign. The other system that you have is called the adaptive system or the acquired system. And this is what develops over your lifetime. This develops when you get exposed to things, when you get vaccinated, you make memory to these things. It's very specific. It remembers specific things you come in contact with during your lifetime. And then when you come in contact with it again, you fight it and you beat it. Okay, and so that's the idea. So today we're going to primarily look at the first and second line of defense. So if you look down here, again, these are both innate, these are both non-specific, and again, no memory associated with this, and these are the different things we're going to look at. So it seems like there's a lot of things, but we're going to talk about them pretty quickly. The third line of defense is going to be chapter 15, so we'll wait and talk about that in chapter 15, but that's the acquired or specific immunity as we look at it. All right, and again, we've already talked about this, so I'll go through this real quickly. But we have the first line. These are your mechanical barriers. These are any things that are found around the portals of entry. So you remember we talked about in chapter 13 how things can get in. When things get in, we need a way to stop them from getting in and causing attachment. So if we can stop them when they come into us, that is our first line of defense. So think about any openings that you have. We have lots of mechanical barriers that keep these things out. The second line of defense sits underneath all the tissues. And so it's kind of, once that first line of defense has been penetrated, the second thing that the microbes want to do is attach. These guys are ready to attack when the microbes want to attach. And so we're looking at now inside the tissues, surrounding the tissues, in the connective tissue, the fluids, things like this. That's where the second line of defense is. The third line of defense is going to be in the cells, in the tissues, and everywhere else. But again, this is going to be the very specific area, and we're going to attack this head on with these specific cells, the B and T cells with antibodies and T cell receptors, and again, very specific. And we'll look at that in chapter 15. All right, let's move on. So first, talk about the first line of defense. So this is keeping you healthy from the outside world. So let's take a look at what is the first line of defense. So the first line is really any mechanical barrier that we have around the portal of entry. And again, we've talked about the portal of entries. These are things like your nose, your mouth, your eyes, your ears, your skin. Those are your portals of entry. And so we have protective mechanisms around those areas to keep the microbes at bay, or at least keep them from getting in any farther. Okay, and so our skin and mucous membranes, the respiratory, urogenital, and digestive tracts are really the main mechanical barrier. The outermost layer of skin always sheds itself after a few days. And so again, we lose that top layer of skin all the time. And that's one way that we lose a lot of our microbes that get on us, or we touch, or we grab, and those things will leave once we shed our skin. Okay, so those are the transient ones. We also have the flushing effect of the sweat glands. So when we sweat, not only does it cool us down, but it helps wash away the microbes as well. Okay, the blinking and tearing up of our eyes. If you know, anytime you get anything near or remotely close to your eyes, you instantly start tearing up. And that is a natural reaction because that's a way that we clean things out. And so those tears have enzymes like lysozyme, which will break down any microbial matter that gets into our eyes. And again, that flushing, the constant tearing up is a way to wash those eyes clean so that they're nice and not sterile, but at least microbe free in that case. Okay. And then the stomach acid is another huge one. Again, a lot of our systems are tied to the stomach acid. So anything that we swallow goes to our stomach, and that acid pretty much kills most things. Now, there are some things that can make it through that stomach acid, but they develop mechanisms to get past it. So, but normal, everyday, run of the mill bacteria and viruses cannot tolerate the pH of that stomach acid. So, that is a huge barrier to getting inside of us as well. Okay, so those are some of the first line of defense. Now, some other things you think about any opening that we have to the outside world, we also have mucous membranes associated with those. And again, those are sticky tissue areas where things will land and collect. And so they land and collect, and that there's a reason for that is that eventually we get rid of those things. So we either break off those layers, we swallow those layers, we do those different things. And so it's really a trapping mechanism. So that's what those mucous membranes are there for. It's not just so that you can cough up wonderful phlegm or have crusty boogers or anything like that. The reason for all that stuff is to trap microorganisms so that eventually we can get rid of them either by sneezing them out, coughing them out, or swallowing them. Okay, and so that's the end. Also, nasal hairs, the cilia and our trachea are also responsible for, again, filtering the air that we breathe. Also, the cilia helps filter out our lungs, all the stuff that we breathe, and that passes it up so that we swallow it. We swallow it in the middle of the night, and then we pass it through our digestive system, which the acid will kill everything. So that's kind of how those guys work, and how we see these mechanical barriers all working together. Okay, some other things that are non-specific that are chemicals. We think of sebaceous secretions, so the oiliness of our skin, there's a reason for the oil. Again, it impedes microbial growth. Lysozyme, I talked about that in our tears. It's also found in our saliva. It helps break down bacteria. The high lactic acid and electrolyte concentration in sweat. So sweat is good. It's antimicrobial because the high salt concentration kills a lot of the bacteria that gets on our skin. Again, the skin's acidic pH also plays a role. So only certain bacteria that can tolerate a high acid concentration can actually live on our skin. The hydrochloric acid, again, we've discussed that. The hydrochloric acid, very strong pH. Again, strong acid destroys most vegetative bacteria. Digestive juices and bile in the intestines. Now, this is closer to a pH of 7. However, these have microbistatic properties, which will actually limit the number of microbes that grow. The fluids like semen have antimicrobial chemicals added to it, so you're not passing certain bacteria along. And then again, the vagina itself has a very acidic pH, which can be affected by, again, hormones, menstruation cycles, and things like this. So again, we see different populations during different times of the month, different years, and things like this. And that's all due to the chemistry of the female at the time. So that's kind of how that works. Okay. So lots of mechanical barriers that keep things from actually getting into our system. So you think of any opening to the outside world, there's typically a mechanical barrier there to block these things from coming in. Okay. Now, another thing that doesn't get discussed a lot are genetic defenses. Now, some people are genetically more viable than others. And again, there is a reason, or there is an idea of the good genes survive out, because some people are more resistant. And this could be due to not having certain receptors that are there. So some people might look at it as a mutation. However, this could be a good mutation where if you don't have this receptor, the virus can't bind, the virus can't get in. So now you're protected. So you don't have that way for the virus to get in. So there are some situations. Good example of this, HIV, there's about 1% of the population that can't get infected with HIV because they lack the right receptor for it to bind in. The receptor that they have that the HIV uses to get into the cells isn't working as well as a normal receptor does. And so some would call that a mutation that could be bad, potentially bad if it's an immune system. However, it actually is potentially good because it prevents you from getting HIV. So that's something that we kind of look at and sometimes we kind of push off. But there is truth to the matter that some people just in their genes alone seem to do better than other people. And you've probably seen this in your everyday life. You see some people that seem to be sick every time, every time you run into them, they have a cold. And other people never get sick. And some of that has to do with their genes that they have. So they just have the right combination to be healthy all the time, and that microbes just don't affect them as well. So again, it does play a huge role. So sometimes it's the genes you get from your luck of the draw, the genes you get from your parents as well to determine your health. Okay, so which of these is not an example of the first line of defense? Which one did I not talk about? Is it the white blood cells, the lysozyme, the hydrochloric acid in the stomach, the sneeze reflex, and the cilia in the respiratory tract? What did I not talk about at all in this case? Okay, if you said white blood cells, you'd be correct. So we haven't discussed the white blood cells. They're going to be in the second line of defense. These sit in the tissues and the surrounding tissues ready to attack if things get through that first line of defense. So if you said white blood cells, you're paying attention. Good job. Okay, so let's move on to the second line of defense. And again, these are going to be associated with the inner tissues. So if microbes somehow make it past our first line of defense, they get entered in, maybe they get through a cut, or we, swallow them in and somehow they get in, or we breathe them in and they get into our lungs. What is our second line to take over for where the first line left off? And so that's what we're going to look at. And again, there's lots of different things that play a role here. Okay, and again, it's a large, complex, diffuse network of cells, fluids that penetrate every organ and tissue. So it kind of surrounds all the tissues and organs in our body. And again, we have really four main classifications or subdivisions of these things. The first one is a very long, hard word, which is called the reticuloendothelial system. Try and say that 4 times. That is basically all your connective tissue. So you have cells that sit in the reticuloendothelial system, and those cells are ready and activated, ready to attack when anything comes in. Think of right under the layer of your skin, you have a lot of connective tissue in that. So if microbes break through a cut in your skin, you have cells that are just waiting there, ready to go after these pathogens that have made it through the skin. Okay. Likewise, in tissues surrounding the heart and other organs, we have cells that are ready to attack if microbes make it into that area. And so that's one of the things. Another one is the extracellular fluid. The extracellular fluid is a big important part. Again, that's all the fluid that leaks out of our vessels. This contains a lot of white blood cells that then go and attack and find the foreign material that isn't supposed to be there. Obviously, the bloodstream plays a huge part of our second line of defense because that carries all our white blood cells to the spots that they need to go. And then we're going to talk about the lymphatic system. Now, some of you may know a lot about the lymphatic system. Others may have never heard of the lymphatic system before. And the lymphatic system is actually a third set of vessels that we have in our body that carries all that extracellular fluid that leaks out back into circulation. So we'll talk a little bit about that. We'll talk about lymph vessels or the lymph channels, and then we'll talk about these little areas that look like these tiny little beans called lymph nodes and why those are important. Think of security checkpoints, and that's what those things are. We'll look at that as well in the second line of defense. Okay? All right, so let's look at these again. We have the reticuloendothelial system, the extracellular fluid, the bloodstream, and the lymphatic system. You can kind of see all of them working in concert here. So if we think of the reticular fibers, these are the fibers that connect all the organs together in that. And again, you have a lot of white blood cells that sit in those fibers waiting to attack if something comes in. We have the extracellular fluid, which is again outside the cells. It kind of bathes the cells. And again, that comes from the vessels. The vessels are leaky, those things come out, and this fluid protects and bathes the cells in all these different tissues. Okay. You also have the white blood cells. You have the bloodstream. Again, the bloodstream brings all the cells, all the fluid to those sites, and so that's important. And then in here, it really doesn't show you, but we also have lymphatic channels, the lymphatic system, which brings all this fluid back to circulation. And so there will be, I guess they do show these lymphatic capillaries. These are these yellow things. And again, it brings in the fluid and brings these things back to the heart. And we'll talk about that as well. So there's a lot of things going on. So if you think about it, the blood is where everything originates from. it gets into the extracellular fluid, you get into the reticular system, which is basically outside the connective tissue. That extracellular fluid kind of bathes the reticular endothelial system, and then that fluid gets returned back through lymphatics, through the lymph nodes, and then finally back into circulation where it goes back through and the whole cycle begins again. So that kind of gives you an idea of what this whole thing plays out. Okay, so let's look at the reticuloendothelial system first, or RES. And so again, this is a network of connective tissues. And really, this tissue has a lot of hiding spots for these wonderful white blood cells to go in. Now, we have a wonderful mononuclear phagocyte system, which is basically these cells that like to eat things. And these monocytes basically camp out and hide in these tissues. So if any microbes or foreign material gets there, they're ready to attack. And so whether it's a virus, a bacteria, a fungus, or even our own dead cells, dead and dying cells, they go in and they eat these things up and then spit them out for breakfast. And that's kind of what they do. And that's their idea of again, fighting the second line. Okay. And again, we find these things all throughout our bodies, in our joints, in our connective tissue of our organs, and all these other places. And so these kind of bathe our organs and where we find this second line of defense. Okay, let's talk a little bit about the blood. And again, blood is where everything originates. And again, the blood is a whole blood. When we think about the whole blood, we have the liquid portion and then we have the cells. The liquid portion is the plasma, and the plasma is about 92% water. It has this yellow kind of appearance. And again, if you spin blood down, you can spin it into its different parts. Again, you have the liquid that sits at the top. You get this little fuzzy area in the middle, which is the white or buffy coat, and we'll talk about that. And then you have the red that sits at the bottom. And again, you can see this on the slide here, the plasma, the buffy coat, and the red blood cells. Now, we call it plasma if we still have the clotting proteins in there. And if you take out and you make a clot in the blood, then you call this plasma serum. So serum, the only difference between serum and plasma is that plasma has the clotting factors, the serum does not, because you've made a clot beforehand. But we're not going to worry about that. We're not worrying about blood clotting in this point in time. But again, that would be something if you were going into looking at blood and coagulation and that stuff, that would be important. But for today, let's just concern ourselves with plasma and kind of look at that. Now, the other two parts of the blood, again, the buffy coat is kind of this furry layer that sits in the middle, and that furry layer is all your white blood cells. Again, it constitutes about 1 to 2% of all your blood is the white blood cells. And then we'll talk more about what all those cells are here in just a minute. But those, again, are your immune cells. These are in there for surveillance. They're in there to go to sites where we have infection or to get to sites where they want to hide out. And so we'll talk about that as well. We also, then again, the big portion, about 50% of our blood, is our red blood cells. And those red blood cells are the ones that carry oxygen. Also in the red blood cells, and you can't see them very well, you wouldn't be able to get them out very well, are the platelets. And the platelets are responsible for clotting our blood. Not only do we have clotting factors, but we also have platelets that are responsible for clotting blood and basically filling the holes if you ever have cuts in your vessels and that stuff. And that's again a clotting procedure. So again, that's kind of what we're looking at with the blood. Okay, and when we look at the cells, we talk about hematopoiesis. And hematopoiesis, or hemopoiesis, I guess, but it should be hematopoiesis, is basically the production of blood cells. Where does this take place? It takes place in the bone marrow. The bone marrow is responsible for making your blood. So any component of your blood is made in the bone marrow. And this is found in your long bones. So your humerus, your radius and ulna, your femur makes a lot of your blood cells. And then you can also talk about your tibia and fibula. Those are also very important for making lots of blood cells. Again, the marrow, which is in the middle of the middle of the bone, is where you have your stem cells. These stem cells are undifferentiated cells that can differentiate into any type of blood cell. So they can turn into red blood cells, they can turn into white blood cells, or they can turn into platelets. And so all these different things work out. What we're worried about are the white blood cells, so we're going to focus in on those first, and then we'll look at the erythrocytes and the platelets here in just a minute. So the leukocytes are your white blood cells. We've already established that, and you have two types. You have the granulocytes, the ones that look like they have granules, and then you have the other ones, which are the agranulocytes, so they don't have granules in them. And so we'll talk a little bit about these different ones here in just a second. So granulocytes are the ones that have granules. Agranulocytes are the ones that don't. Again, I talked a little bit about stem cells. This would be the stem cell sitting in your bone marrow, and again, it can go off into any direction, making any blood. So we call this a hematopoietic stem cell, meaning that it can make any of the blood cells. Now, some of them will go in one direction, and they will go and make your red blood cells. Okay, some of them will go into another direction and make platelets. Some of them will go down and make the granulocytes. Others will go down and make monocytes, which is an agranulocyte, and then others will go and make your B and T cells. So again, think of it this way. All your white blood cells, all your red blood cells, and your platelets come from one master stem cell. Now, I'm not saying that there's just one stem cell that does it all, but there's one type of master hematopoietic stem cell that you have in your bone marrow that produces all your different types of blood cells. And so when you go on like things like cancer treatment drugs and things like this affects your blood because your blood are rapidly reproducing all the time, your white blood cells, your red blood cells. And what are the main problems when people go on cancer drugs is that they will go anemic because they don't make enough red blood cells, and then they have a hard time with infections because their white blood cells are affected. And that's because they all come from the same type of stem cell. And so that can be an issue. So kind of think of that. That's just kind of extra material just added in, but that's why you have so many problems when you go on cancer treatment drugs and that, because it does affect all these rapidly dividing cells. Again, one stem cell controls all your different types of blood cells. Okay, so we first talked about granulocytes. Now the granulocytes include things like neutrophils. And neutrophils are about 55 to 90% of all your white blood cells. So if you go back to that tube of blood, and you remember I showed you most of it was plasma on the top, some of it was the red blood cells, or it was about 45% plasma, 55% red blood cells, and about 1% of the white blood cells, that buffy coat. And out of that 1 to 2% of the buffy coat, 55 to 90% of those are actually neutrophils. And those neutrophils are basically these indiscriminate cells that just go and eat. Their job is to go around and chop things up just like a Pac-Man would on a video game. So it just goes around, chop, 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 looking for pathogens, okay? Looking for pathogens or things that shouldn't be there. And so their job is to go find foreign things and eat it. And so that's the neutrophils, okay? So that we just associate them with phagocytosis. Another type of cell that you have, and it's a very small percentage of cells that you have, are about 1 to 3% of, again, that 1% of white blood cells are called eosinophils. Now, eosinophils are specifically against eukaryotic parasites. So if you get infected with a eukaryotic parasite, say you get a protozoan, so you drink dirty water and you get a protozoan, or you get a worm like a tapeworm or something else, your eosinophil count will actually go up. And that's one of the ways that doctors can figure out that you have some type of eukaryotic parasite, because if they see on your blood count that your eosinophils are high, that says directly, aha, you guys have a parasite. So you need to figure that out. So that's why it's important to do these white blood cell counts to kind of figure out what type of infection is going on. The type of white blood cell that's raised up can tell you a lot. Neutrophils go up when you have a bacterial infection. Eosinophils go up when you have a parasite infection. Now, the basophils are even a smaller percentage of cells, and these are really geared to, and again, there are some reasons why we have these cells, but we're really going to talk about these guys is in chapter 16 when we look at allergies. You can blame the basophils for your allergies because these are the guys that release their histamine and leukotrienes and everything else and cause you to have an allergy reaction, and that can be a bad thing. And so that is what causes you to have those allergy responses. Those are the basophils. So the basophils are why you get the allergies. And we'll talk about those again in chapter 16 and how you get an allergy attack and response. Okay? All right. Now we have some that are agranulocytes. And again, we have the B and T cells, and this is about 20 to 35% of our white blood cells, again, out of that 1%. And these B and T cells, again, are for the third line of defense. So we'll talk more about these in chapter 15. We'll spend some time talking about how they get their receptors, what the receptors are, and how they get specificity. And so, again, we think of B cells and antibodies, T cells and their T cell receptors. And again, this is their specific thing. And we'll look at this again in chapter 15. The last one of the agranulocytes are the monocytes. So these are very similar to what the neutrophils are. Okay, neutrophils, remember, are the little Pac-Men that go around and eat. Well, the monocytes will do the same thing. They'll go around and eat pathogens. However, their responsibility is then whatever they eat, they're going to show it to the B and T cells. So I like to call the monocytes or the macrophages the link between the innate immunity and the adaptive or acquired immunity, because these cells are responsible for eating things up in the innate system, but then they turn around and then they present to what they ate to the T cell and say, look what I ate, you need to do something about this. So they're kind of the early warning to get the acquired immunity started. Now there's always a delay with the acquired immunity, and again we'll look at this in chapter 15. And the reason for that is that the macrophages have to eat something first in order for them to respond to the T cells. So it takes some time for the system to get going. And so that's why you have that delay. And so monocytes are very important to link the innate immunity to the acquired immunity. And again, we'll look at that link again in chapter 15. All right, now I mentioned briefly about the erythrocytes. These are your red blood cells. And again, red blood cells lose their nucleus. And the reason for that is because they don't need to replicate anymore. Once they get to the red blood cell stage, all they're going to do is carry oxygen, and that's their lifespan. Once they've done that, the cells die. They go to the spleen to die, and that's it. Okay, and so these cells don't replicate once they get to that stage. And so since they don't need to replicate anymore, once they get to the erythrocyte, they lose their nucleus. So these are kind of the donut-shaped cells. When you look at a blood smear, you can see down here, these guys are all the red blood cells. They have no nucleus. When you look at white blood cells, they stain purple because they do have a nucleus. Platelets also stain a little purple because they have little splotches of nuclei in them as well. Now the platelets are elements to help clotting the blood, and again, these come also from the stem cells. These come from megakaryocytes. These are like pieces of cells that are used to fill up holes when your blood vessel ruptures. And so when you have a cut or an injury to the vessel, these guys respond by plugging up the hole so that you don't bleed to death. And so the platelets are really important for blood clotting, and that's about the extent that we're going to get into today. Now there's some other roles for platelets and that stuff that I could go into and really bore you to death with, but for today we'll just think of them as a clotting mechanism. Okay, they get activated and they plug the hole. Think of it that way. All right, so that's the platelets. All right, now let's talk a little bit about the lymphatic system. The lymphatic system is the third set of vessels that we have. So we have the arteries, which take blood away. We have the veins that bring the blood back. And then we have the lymphatic system. The lymphatic system is a series of vessels that carry fluid that leaves the vessels. And again, I told you the vessels are leaky, and carry these vessels back to the heart. So it brings the fluid back to circulation. And so it provides an auxiliary route to return extracellular fluid back, acts as a drain-off system for the immune system, and again, we'll talk about why that's important. And again, what it does is it renders surveillance for all the fluid that goes around. So there is a way to monitor what's in your system. And so the lymph system is very important for surveillance and looking at things that get out of the blood and are actually in your tissues. So if it gets under your skin, those things get picked up and get brought back and surveyed so that they know exactly what they're dealing with. And we'll talk about that as we go through the lymphatic system. So the lymphatic system is necessary for bringing fluid back to the heart. Okay, now again, lymph is any extracellular fluid that leaves the vessels, and it kind of gives you this yellowish green color. And again, it's very similar to what plasma is. It's got some components of blood. Again, it's made up of water, dissolved salts, and some proteins, and so that's what gives it its color. And again, it's just any extracellular fluid that leaves the blood vessel. Okay, what does it do? It transports white blood cells, fats, cellular debris, and infectious agents throughout the system. So one of the things I didn't really talk about is that, again, organisms can use this as basically a highway system to get anywhere in your body. So they can use the lymph system to go from one place to another. Another place that we see this is with cancer. So cancer is notorious for using the lymph system from going to one organ to another. So a lot of times when you hear about breast cancer and that, what they'll do a lot of times is remove some tissue for a biopsy, and they'll also look at the lymph nodes that are found in the armpit. The reason for that is breast cancer especially, likes to use the lymphatic system to go to other parts of the body. So what they're doing is they're actually looking at the lymph nodes there to see if any cancer cells have actually made their way there. If they have, then they know that the cancer is trying to spread, and that allows the doctors to know, ooh, this is something we need to really be aggressive about. And so that's one of the issues with the lymphatic system. So that's another thing. Think of this highway system where things can utilize it to get around the body, and we'll talk about that as well. Okay, so lymphatic vessels are capillaries that permeate all parts of the body except the central nervous system. However, that's not true. They are actually finding lymphatic system in the brain as well. So this is old research. The new stuff that just came out last year shows that we actually have lymph vessels in our brain. Okay, bone, placenta, and thymus. It has thin walls that allow for that can be permeated by extracellular fluid, so the fluid can come into these vessels. And anytime we move lymph fluid back to the body. Okay, and again, the function is to get the lymph back into circulation. Any fluid that leaks out has to get back into circulation somehow, and these vessels allow it to go back into the system. Now, you may notice in this picture here, you see these little bean-like structures. These bean-like structures are called lymph nodes, and lymph nodes are basically checkpoints to see what's going on in the fluid. So basically, these lymph nodes can look, survey the fluid that's going by, and say, do we need to do anything about it? Because a lot of times microbes will be found in this fluid because it's got into the extracellular fluid. It then gets into the lymph node, and you actually have white blood cells, macrophages, B and T cells that are all sitting in that lymph node ready to attack. And they're essentially sitting there waiting for the right thing to come along so they can go and attack the thing that comes by. So think of it as like a checkpoint or a border crossing type of thing. This fluid gets checked, and if it, if they find something, it's going to react. And you know when these things react because then the lymph node swells, and that's what you feel in your neck. Okay, so that's the function of the lymph nodes essentially. Okay, so again, the extracellular fluid leaves the vessels, and again, a lot of times through the capillaries, the fluid makes its way back into the lymph system. That lymph system will take it back. Now, again, I talked about the lymph nodes already. These are these bean-shaped, I wouldn't say organs, but these nodes that, again, the fluid comes in, and you have these packed with different types of immune cells, B cells, T cells, and macrophages. And these guys come in and they survey the fluid, and if they find anything, they get activated and they start to attack what's in here. And again, they send out the warning signal. The rest of the fluid makes its way back, and eventually it will feed back into circulation. And that's the idea of the lymphatic system. Okay? All right. Now we have two types of lymphoid organs. We have the primary lymphoid organs and the secondary lymphoid organs. The primary lymphoid organs are things like the bone marrow and thymus. So when we think of primary, think of where white blood cells are formed. Okay, and white blood cells are formed in the bone marrow. All our white blood cells are formed there. And then the thymus is an important organ for or gland for maturation. Okay, so the bone marrow allows for the B cells to mature and get the receptors. The thymus, again, the T cells are made in the bone marrow and then go to the thymus to get receptors, and that's important. And so you get these T cells that now have receptors on them and they're ready to go. And so those are both primary lymphoid organs. Now, when you're a baby, your thymus gland is very, very large. But as you get older and you get into puberty, it shrinks. It starts to shrink even more. By the time you're 70 and 80 years old, you have really no thymus gland left. And so again, that plays a role in your overall immunity. That's why as you get older and older, your immune system goes away because your T cells aren't being as activated and matured anymore because you're losing that thymus over time. Now, the other organs that we have are the secondary lymphoid organs, and these are wherever our white blood cells go and hang out. And so we have these lymph nodes, again, those are where B and T cells sit. We have our tonsils, which are basically glorified large lymph nodes. We have our spleen, and we have some other areas around our intestinal system that also acts as a lymph system and basically a surveillance system area. And so all these different tissues that have the white blood cells just sitting and lurking are called secondary lymphoids. So we have the primary and then the secondary. And I think the next slide we'll talk a little bit more about that as well. So the primary are your bone marrow and your thymus. Okay, these are where the cells are made and matured. Okay, so bone marrow makes all the cells, but bone marrow matures the B cells. The thymus is where the T cells mature. And again, we'll look at that in chapter 15, but those are the primary. So either made or mature there. Okay. The secondary are where the white blood cells go to hide, and so that's a big thing. So the lymph nodes, the spleen, and these other areas that are around the body. And so again, these are for surveillance, and so they come in and survey the area, and so these are the secondary lymph nodes. Okay, so primary is where they are made and go and mature. The secondary is where the white blood cells go and hang out. Okay. Okay. So we have the T cells. The T cells mature in what? The lymph nodes, the spleen, the thymus, or the gut. Let's see if you can think of that one. The T cells mature in the thymus. So again, we talked about that. That is a primary lymphoid organ. The T cells get the receptors there and they mature at that spot. So that's where you're going to see the T cells. Okay, T cells mature in the thymus, and that's actually why they're called T cells, because of the thymus. Okay, so if you got thymus, good job. You're paying attention to the second or the primary lymphoid organs. Okay, so now the rest of the lecture, we're going to spend some time just talking about how the white blood cells do what they do. So we're going to look at inflammation, phagocytosis, interferon, and complement. So we'll look at those four things, and this is basically how the white blood cells do what they do. Okay, so the first one is going to be inflammation. Now, inflammation is a response. This is basically whenever anything gets in and your white blood cells attack. So think of this as the innate immune response. Okay, this is a response that takes place. These are the classical signs. You get redness, warmth, swelling, and pain. The reason for this, the redness is due to the vasodilation. So this is where the blood, again, more blood to the injury. The blood then leaks out. Okay, so that's where you get the redness. The warmth is again from the vasodilation, more blood to that area. The swelling is due to the leaky fluid. The fluid again contains the white blood cells that are going to help fight the infection. And then pain is due to the swelling because it tickles the little nerve endings and causes the pain. And so you get those all associated. And so you've probably seen, different examples of this. So if you prick yourself and let's say this little thorn, the thorn cuts you, you get more blood to the site to, again, take care of the wound and that stuff. So you get the redness and the swelling, or the swelling here, the tumor. Okay, and then you get the rubor and color, which is basically the redness and the color and the heat. And then you get the pain, which is associated with, again, the tickling of the nerve endings due to the swelling. And so those all play a huge role in the inflammatory response. So visually, what happens in inflammation? So let's say you get poked by either a needle or a nail or a pin. And so what happens is these guys are coated with bacteria. We know this. Again, you guys did this in the lab looking at all the stuff that's out there. The pin comes in, breaks the skin, and it brings a nice dose of bacteria into the body. The bacteria get in, you get a rupture of the vessel. Okay, so the first thing you want to do is make a clot. So this is where your platelets, your other clotting factors all play a role so you don't bleed to death. So that forms a clot. Meanwhile, your neutrophils are the first responders. They go in and they're saying, oh, injury, let's go eat, 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 eat. And so what they're doing is they're there to eat. And so they leak out of the blood vessel and they go and eat all these wonderful little green bacteria. So that's what you see. Now you get swelling associated with that as well. So this is not only the fluid that leaks out of the vessel that could be leaking out because of the injury or just in general the inflammation, but you also have cells, you also have pus formation, and pus is basically the waste product of the neutrophils. So neutrophils are eating up and basically digesting these bacteria, and then this is the waste that is from the digestion of the bacteria. So pus means that your immune system is working well and it's cleaning that area out of the bacteria. Now, eventually what comes in are these very large macrophages, and again, these are going to be important to stimulate the T cell and B cell response. So they come in and kind of finish up the job that the neutrophils leave off. And so these kind of guys kind of come in and take the credit at the end and clean up the system. They get rid of any remaining bacteria and say, hey, T cells, let's respond to this. Let's make memory to this. And so they'll eat it and then they'll present it to the T and B cells and say, let's make some memory so that we know that the next time we get this, we can fight. And those are the major events of inflammation. So you get the injury, you get the swelling, the redness, and warmth due to the blood leaving the vessel. The swellings due to the fluid that's leaving the vessel, along with the cells eating. Some pus formation is due to the digestion of the bacteria due to the neutrophils. And then finally, the pain is associated with the tingling of the nerve fibers. So all these different things are going on during the signs of inflammation. Okay, and that's really what happens during an injury. All right, so some other things that the white blood cells can do is even when you don't have injury, is these things can leave the blood vessels on their own. So they don't need an injury to the vessel in order to leave. They can do what is called diapeditis, and diapeditis is basically the movement out of blood vessels. And you can see here, these wonderful little white blood cells down here can actually either work their way through either the middle or basically the openings are in between the cells. They work their way in and kind of force their way in and work, and you can kind of see this happening, and this is called diapeditis, and force their way through the cells. They actually even can go through the cells too. There's actually some research that's been done that shows that white blood cells will actually even go through the middle of cells sometimes if they have to. Okay, so this shows you that the blood vessels, even though they look like tubes, are not a solid structure, that things can actually get through. And again, what brings them there is the chemotaxis. So cells that are already present will release these chemicals to say more cells come to the area and take care of this infection. And so you get this going on all the time. And so it's kind of an early response warning, and that brings more cells, and more cells come in. And finally, once the infection clears, you're free of the pathogen. Okay, and so that's what goes on in these situations. Okay, now another thing that happens is fever. Fever is a normal response to when you have an infection. And so anytime your body knows that it has foreign material in it, it will send a signal to the brain to say, well, let's warm it up. And so what the warming is supposed to do is remove the pathogens from there. And anything that causes or triggers fever, we call pyrogens. So think of fire. Pyrogens stimulate heat, and that is due to the chemical response that triggers the hypothalamus to turn or reset the thermostat in your body and bring up the heat. Now we have both exogenous pyrogens that are found on infectious agents, so sometimes the microbes themselves will trigger a fever to occur, and other times we have cytokines and other chemicals that will be released by our immune system to say, hey, let's turn up the heat a little bit to help kill these microbes. And the reason why we do this is that we have some benefits to fever. So a lot of times we associate fever and think bad. Ooh, we got a bad fever, we need to take care of it. But a low-grade fever a lot of times are actually very beneficial, because what it does is it helps control the spread of the microorganism. So one, it can inhibit multiplication, so that's always good to keep the bacteria from growing. It impedes nutrition, so if you keep the nutrition from the bacteria, they have a hard time multiplying and growing, so that's good. And it can also increase the metabolism and stimulate your immune system. So low-grade fevers actually can be a good thing. And so I don't want you to assume that all fevers are bad. Okay, so that's the one word of advice I can give, is that fever sometimes is not a bad thing. Now, obviously, if a fever gets above 103 to 104 and 105, that's when you really have to be worried. But if you have a low-grade fever like 100 or 101, those are actually good things. That means your body and your immune system is working and it's fighting off infection. So again, the next time you have a low-grade fever, you might want to restrain yourself from getting the ibuprofen or the Motrin and letting it take its course. Sometimes when we take medication, it only is prohibiting the effects of the healing process. So just remember that sometimes that a low-grade fever can be actually a good thing for you. All right. So one of the last things we'll look at is phagocytosis, and again, this is to eat up foreign material. And so I have a couple videos to show you not only phagocytosis, but the last thing in complement. And what phagocytosis is basically to survey the tissue, find these things, and eliminate them. Okay, so look around for these, again, microbes, particulate matter, and dead and injured cells, then to ingest them, eliminate these materials, and then take immunogenic information and pass it on. So this is specific to the monocytes. So you remember the monocytes are that link between the innate and the adaptive immunity. And again, what they do is they eat things, and then they take the information and then present it to the T and B cells. So that's specific to the monocytes. These are more for the neutrophils and the monocytes up here, 1 and 2. And neutrophils, again, just eat and just digest, and that's all they do. They don't present. And so the monocytes are the ones that present the material. Okay, so let's watch this video on phagocytosis and take a look at what these things really are, okay? And what happens in this process.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
Okay, so again, this video is showing you a little bit about how the bacteria eats the stuff. It brings it in through endocytosis, digests it through the lysosome. So we talked about the lysosome in chapter 4. Those are those enzymes that are in vacuoles that get released and digest. And again, this is a neutrophil since it just kind of uses the exocytosis to eliminate the pieces. And these pieces end up forming the pus. And so that's part of the pus, not only the extracellular fluid, but any of the digestive waste products that you see from the neutrophils themselves. Okay, so that's what we're seeing with phagocytosis. Okay, now again, some of the cells that are able to do this include the neutrophils and eosinophils. Remember, the neutrophils, they're primarily for bacteria. These go in and eat up bacteria, dead cells, and other things. And again, they eat them, process them, and then spit them back out, basically, after they're done digesting them. Eosinophils are for the eukaryotic parasites. We talked about that. Again, you'll see when we have eukaryotic parasites, this number of eosinophils actually goes up. And that's one measure to look at. Now the macrophages, like I said, these are the specific, these are the monocytes, these are the guys that once they leave the blood vessel, they start to eat things up. And these guys, once they eat them, they will digest them and then they will present them. And so this is the important part, that they will eat, digest, and then present to the adaptive immunity, to the T and B cells. So again, we'll look at this again during chapter 15, looking at how the B and T cells get presented. And we'll talk about what the role of the macrophages and dendritic cells. So these are both the macrophages and dendritic cells. If you're wondering what those are basically monocytes that have left the blood vessel. And so they get different names. And again, they get a little bit different structures. They have some different receptors and things like this, but I just wanted you to be aware of those things. Okay. Okay. Now here is phagocytosis. I think this is a really cool picture. This shows you some neutrophils actually eating up the bacteria. So that's kind of cool in that. And what's going on here is, again, basically the neutrophil will find the bacteria through chemotaxis. It will bind the bacteria up in the receptors. It will bring it in, and here comes a lysosome. So that's called a phagosome. That's kind of like the stomach of the cell. These lysosomes come in, release these enzymes, digest the bacteria, it gets the parts, and then the release of those residual debris. And so that's where those things go. And that's primarily the process of phagocytosis. Okay, so the phagocytosis is bringing these cells in, digesting, and then releasing them out. And this would be a neutrophil since it's not presenting those cells. Okay. Now, interferon is an interesting chemical, and we talked about this during the antibiotics. So it was one of the last things we looked at during the antibiotics, if you remember. And this is a natural chemical produced in response to infection. And a lot of times we see this in response to viruses. And so this is kind of when a cell gets infected with a virus, this kind of sends out the silent alarm to other cells saying, hey, we need to do something. We're getting infected with viruses here. And so interferon does a really good job of turning on these other cells. And so it's a chemical that gets released. It turns on other cells. You can see there's different types that turn on different types of cells, and these things respond to the infection. And so this is kind of the silent alarm to say, hey, I'm being infected by a virus. Let's do something about it. Primarily, we're talking about phagocytosis. This is the bacteria and other things getting eaten up. Viruses, on the other hand, we need to deal with them in a different way because they actually infect cells. And so we'll look at this again. Chapter 15 plays a huge role with turning on the T and B cells and how to fight these viruses inside of cells. But this is one of the ways that we do it, is with interferon. And again, we're seeing this as well with cancer treatments as well. Again, it's a natural molecule that we can add to turn on the immune system. And so this is being used also for cancer, because the immune system can help fight cancer as well. So this is one of the new treatments that we see. Okay, the last thing we're going to talk about is complement, and complement is a series of 26 proteins that react to different signals in the body to say, okay, we need to destroy these pathogens. And so a lot of these things get turned on through different mechanisms in the body. Some of these are through innate mechanisms, and others are through the adaptive or acquired mechanisms like antibodies and antigens and that stuff. And so, again, I'm not going to expect you to know all these different proteins and how these all do. What I just want you to know is what complement is. Complement is a series of blood proteins that interact with the pathogens and destroy pathogens. Not only do they turn on the immune system, but they also can destroy pathogens by basically putting holes in the pathogens, okay? And again, there's different pathways. I'm not going to expect you to know the different pathways and the proteins and that stuff. I think in A&P, some of you guys may have been bounced over the head a few times about learning complement and what it does. But in here, all I care about that you understand is what is it for, okay? So if you understand that, what it's for, why it's there, I'm happy. All right, and I got a video just to show you the complement pathway here.
Complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different foreign molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are C1 through C9, named in the order that they were discovered, not in the order in which they function. The complement cascade can be activated by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated C1 then cleaves C2 into C2A and C2B, and C4 into C4A and C4B. C2B and C4B combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3A and C3B. In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3A and C3b are constantly being formed from C3, but without activation they are soon destroyed. C3b reacts with the proteins factor B, factor D, and properdin to form a complex called C3 convertase, which cleaves C3 into C3A and C3b. Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3A is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3A and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have a binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properdin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5a and C5b. C5a enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components including C6, C7, C8, and C9 to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
Okay, so this gives you an idea of what takes place with complement. And again, I'm not going to expect you to know all the pathways and everything else. What I really want you to get out of it is that you know that we have a system of proteins in our blood that actually will help fight against one, turn on the immune system either by the stimulation of inflammation or opsonization. Again, we'll look at that in chapter 15, or being able to plug holes into or poke holes into the bacteria cells. And so that's really what I want you to get out of it. Again, don't worry about memorizing all these proteins and everything else. Okay, so if you understand what complement is, I'm happy. So that's good. All right, so let's finish up with a concept check. So which of the following defenses is most likely to be active during a viral infection? So I talked briefly about this. What do you think it is? Is it inflammation, fever, interferon, or complement? Okay, if you said interferon, you'd be correct. So that was the chemical, like I said, that is kind of the silent alarm. So when cells start to get infected with viruses, they can send that silent alarm out to other cells and stimulate them and say, hey, I've got a virus inside of me. Let's do something about this. And so interferon is a great component of that. It's kind of like the self-sacrifice to save the rest of the team. So this cell will die eventually because it's getting infected, but it will send out a signal to the rest of the cells either to stimulate the immune system or the other cells to block the virus from actually getting in. So very good if you got the interferon. All right, so let's summarize. So immunology is the study of the immune system, and again, that's looking at the different components of the immune system and what's made up of all those different parts. And again, we talked about how the immune system surveys, finds, and destroys. So surveys, finds, and destroys, if you know that, that's pretty much immunology, and it's a study of looking at those things. And I could spend days and days and days talking about the immunology. Okay. What we looked at today was the first line of defense. This is the first part of the innate. This is the mechanical barriers, and think about anywhere that you have open, portals of entry, you're going to have these mechanical barriers ready to basically protect those portals of entry from getting infections. And so again, these are the structures and chemicals that keep foreign invaders out. And this includes the skin, mucous membranes, tears, wax, sweat, any of those things, any of those chemicals that are produced to fight off microbes. The second line of defense is the cells and inflammation. Again, we talked about it. It's really all the things surrounding the tissues and the organs. And the second line of defense primarily looks at the white blood cells, and those are the granulocytes and the agranulocytes. And again, we don't think about the T and B cells yet. That's going to be for the next chapter. But everything else. So the monocytes, the neutrophils, the eosinophils, and the basophils are all important cells for the second line of defense. The lymph and the lymphoid tissue, again, very important for bringing back the extracellular fluid that leaks out of the vessels. Again, goes through the lymph nodes where surveillance takes place. Remember your primary versus your secondary lymphoid tissue. Primary is where the white blood cells are formed or mature. So that's the bone marrow and thymus. And then all the other tissues like the spleen, the lymph nodes, the tonsils, and all those are secondary because that's where the white blood cells go to hang out. Okay, inflammation, the four things are redness, swelling, heat, and pain. Those are the typical signs and symptoms that you are associated with inflammation. So if you remember that, you're good. And then we talked about phagocytosis, which is basically eating the foreign material and then spitting it out. If you're a monocyte, you actually take those particles and you present them to the T cells. And then complement is a series of blood proteins that will respond to infection in the body. And so that looks at the second line. Now, in this last slide I have here is just showing you a little bit about the third line of defense. Again, what separates the third line of defense from the first two is that one, it's acquired. This is something that develops over a lifetime. So it's what you're exposed to, either naturally or through vaccines that tell you what type of immune system you develop. And again, you develop specificity, and so it's what you see. So what you see is what you're going to respond to. And again, this deals with the B and T cells. We'll talk about memory and all these other things in the next chapter. Okay, so we've come to the end of chapter 14. If you have any questions about anything, please don't hesitate to ask. Send me an email, stop by office hours, send me a text, whatever you need. I'm happy to answer at any time. And I'm glad that you're enjoying the videos, and I'm glad that you're watching, and I will see you next time.